The southern Appalachians in autumn look like something the earth invented to make people stay. Ridge lines stacked to the horizon, each one a shade darker than the last. Hardwood forests burning gold and copper in the October light. For thousands of years, this was the heart of Cherokee country. Western North Carolina, eastern Tennessee, the high valleys of northern Georgia. And for most of that time, the story seemed simple. The Cherokee were a southeastern people. Their roots were here, in these mountains, in the rivers that carved through them, in the mounds and village sites scattered across the Blue Ridge. That story made sense. Until you looked at the language. Cherokee is an Iroquoian language. The rest of the Iroquoian family, the Mohawk, the Seneca, the Oneida, the Tuscarora, they all lived over 800 miles to the north and east. Great Lakes country, upstate New York, the St. Lawrence Valley. The Cherokee sat alone in the deep south, speaking a tongue that had no business being there. Linguists noticed this early. By the 19th century, they were already asking, how did an Iroquoian-speaking people end up in Appalachia? For generations, the answer was vague. Maybe a prehistoric migration, maybe a very ancient split. The details stayed murky because the tools to resolve them did not exist yet. Archaeology could map settlements and pottery styles. Linguistics could estimate divergence dates, roughly 3,500 to 4,000 years, which suggested the Cherokee separated from Northern Iroquoian groups during the late Archaic period. But neither field could tell you who moved, in what numbers, or what they carried with them. DNA changed that, and the answers it provided were not simple. Here is the part most people do not know. When geneticists first began cataloging Native American mitochondrial DNA, the system they built was clean, almost elegant. 4 founding haplogroups, labeled A, B, C, and D, each traceable to East Asian source populations, each consistent with a Beringia land bridge crossing during the last ice age. Roughly 97% of all Native American maternal lineages fit into these 4 boxes. The model worked. Papers were published, textbooks were printed. Then the remaining 3% started causing problems. A 5th lineage kept showing up. Haplogroup X, and Haplogroup X did not behave like the other 4. Haplogroups A through D all have clear parallels in East Asian populations. That is what you would expect if every founding lineage crossed through Beringia from Siberia. But Haplogroup X, the Native American version, is not concentrated in East Asia. Its closest relatives are in Europe and Western Asia, the Middle East, the Caucasus, populations that have nothing to do with the Beringia crossing, at least not in the traditional model. I need to be careful here, because this is exactly where the story gets hijacked. Every few years, someone tries to use Haplogroup X to argue that Native Americans are really European, or that ancient Israelites colonized the Americas, or some other pseudoscientific fantasy designed to undermine indigenous identity. None of that is what the data shows. I want to be very clear about that. What the data actually shows is more interesting and more complicated. In 1999, 2 teams published landmark studies almost simultaneously. Michael Brown and colleagues at Emory University, and David Glenn Smith's team at the University of California, Davis. Both groups had been tracking Haplogroup X across Native American populations, and both reached the same conclusion. The Native American X lineage, designated X2A, is real. It is not European contamination. It is not colonial-era admixture. It diverged from its Eurasian counterpart at least 15,000 years ago, probably more. That date matters enormously. It means Haplogroup X was already a distinct lineage before anyone set foot in the Americas. It arrived with the founders. But here is the part that connects back to the Cherokee. When Smith's team mapped the distribution of Haplogroup X across native North American populations, the pattern was striking. It was not spread evenly. It concentrated heavily in the Great Lakes region, among Algonquian and Iroquoian-speaking peoples. the Ojibwe, the Sioux, the Nuu-chah-nulth, and it appeared, at lower but notable frequencies, in Cherokee populations, the same geographic footprint that the language pointed to. If you are the kind of person who pays attention when two completely independent lines of evidence converge on the same answer, this is where you should sit up. Because the Cherokee were not supposed to be genetically connected to the Great Lakes. They were supposed to be southeastern, local, Appalachian. If the idea that a single genetic lineage could rewrite a migration story sounds like something you want to follow, this is the channel for that. We go where the evidence goes, even when it gets uncomfortable. 3 years after those haplogroup X studies, a young researcher named Ripan Malhi at the University of California, Davis, was doing something nobody had attempted at this scale. He and his colleagues were assembling mitochondrial DNA data from across the continent, not just from one tribe or one region, but from dozens of populations spanning the Arctic to the Gulf Coast. 1,612 mitochondrial DNA samples from native North Americans. 395 full sequences of the hypervariable segment, each one a thread in a genetic tapestry that had taken thousands of years to weave. It was the largest dataset anyone had assembled at the time. And when the team ran spatial autocorrelation analyses, plotting haplogroup frequencies against geography, the continent organized itself into 3 distinct clusters. Haplogroup. A populations had experienced prehistoric expansions in the Arctic North. Haplogroup B populations had expanded in the Southwest. And Haplogroup X populations, the anomalous 5th lineage, had expanded around the Great Lakes. Picture that result appearing on a screen. A map of North America with colored dots marking haplogroup concentrations. The Great Lakes lighting up for Haplogroup X. Now overlay a second map, one showing the known distribution of Iroquoian languages before European contact. The overlap is not approximate, it is precise. Actually, wait, let me back up here because I skipped something important. The reason Haplogroup X matters for Cherokee origins is not just that Cherokee carry it. It is that the distribution of X overlaps precisely with the region where linguists place Proto-Iroquoian before the Cherokee split away. The Great Lakes, the Northeast Corridor, the very territory that Cherokee oral traditions describe as their homeland before they migrated south. This is not one data point lining up. This is 3. Linguistics, genetics, and oral tradition, all pointing at the same map coordinates. Then in 2011, the evidence got even more specific. At Washington State University, Deborah Lawrence and Brian Kemp were working on something that most researchers would have considered a side project. They had access to skeletal remains from 3 Arikara village sites along the Missouri River in South Dakota, sites that had been occupied between 1600 and 1832 AD. The Arikara speak a Cadun language, a completely different family from Iroquoian, and completely different from Siouan. On paper, there was no reason to expect a Cherokee connection. They extracted mitochondrial DNA from 63 samples. 55 yielded usable data, an 87% success rate that speaks to the quality of the preservation. The 4 major haplogroups appeared at frequencies of 42% for A, 29% for B, 22% for C, and 7% for D. But it was not the frequencies themselves that mattered. It was what happened when the team ran principal coordinates analysis, a statistical method that places populations in multidimensional space based on how similar their genetic profiles are. Imagine each population as a point in a room. Closer points mean more shared ancestry. Farther points mean less. The results were remarkable. The Arikara clustered closest to 2 other populations, the Siouan-speaking peoples and the Cherokee, not close to the Pawnee, their nearest geographic and cultural neighbors, not close to the Mandan, who lived along the same river system. The Arikara's deepest genetic affinities were with groups hundreds of miles away. There is an old and controversial hypothesis in linguistics called the Macro-Siouan theory, which argues that Iroquoian, Siouan, and Caddoan languages all share a deep common ancestor. Most linguists considered it unproven. Lawrence and Kemp's DNA data did not prove it either, but it made ignoring it significantly harder. Think about what that means. Three groups separated by hundreds of miles, speaking languages from three supposedly unrelated families, and their deep maternal lineages told a story of shared ancestry. Not recent shared ancestry. Ancient shared ancestry, the kind that predates the divergence of the languages themselves. What this evidence shows, taken together, is that Cherokee maternal lineages carry signatures of a northern origin, specifically the Great Lakes corridor, consistent with a migration southward into the Appalachians roughly 3,000 to 4,000 years ago. What it rules out is the old assumption that the Cherokee were an in-place southeastern population whose roots were entirely local. The genetic diversity patterns, particularly the presence of haplogroup X and the deep lineage sharing with Great Lakes and Plains populations, cannot be explained by in situ development. What it implies is extraordinary: that a substantial population movement, large enough to establish a language and a genetic footprint in the southern Appalachians, occurred during the late Archaic period, reshaping the cultural landscape of the Southeast long before Mississippian civilization arose. But I should be honest about where the debate still stands, because it would be misleading to present this as a closed case. I spent a long time trying to find a good counter-argument to the northern migration model, and I will say this: the skeptics raise some legitimate points. Mitochondrial DNA is maternal only. It traces one line of descent through mothers. It says nothing about paternal ancestry, nothing about autosomal DNA, nothing about the full genetic picture. A population can carry northern maternal lineages and still have deep local roots through other lines. Admixture is complicated, and mitochondrial data alone cannot distinguish between a full population migration and sustained intermarriage over centuries. There is also a sampling problem that is hard to ignore. Most Cherokee DNA studies have used modern Cherokee populations, primarily citizens of the Cherokee Nation in Oklahoma. But the Trail of Tears in 1838 forcibly relocated approximately 16,000 Cherokee from their southeastern homeland to Indian Territory, killing an estimated 4,000 in transit. 1 in 4 people dead. That is a devastating population bottleneck. The genetic profile of modern Oklahoma Cherokee may not accurately represent pre-contact Cherokee diversity. It might overrepresent certain lineages and underrepresent others purely by the randomness of who survived. And yes, I know that sounds like a technicality, but in population genetics, bottlenecks reshape everything. A lineage that was rare before the Trail of Tears could appear common after it, simply because its carriers happened to survive. A lineage that was common could vanish. I rewrote this section 3 times because the evidence genuinely pulls in 2 directions. The bottleneck is real, and it complicates the data. But it does not erase the signal. It just makes it noisier. The Eastern Band of Cherokee Indians in North Carolina, descendants of those who avoided removal, offers a different genetic window. But that population is small, and sampling has been limited. This is where the story becomes genuinely frustrating, because the evidence we need, ancient DNA from pre-contact Cherokee archaeological sites, barely exists. The technology is there. Researchers have extracted and sequenced ancient DNA from remains across the Americas, including the 12,600-year-old Anzac child in Montana, whose genome confirmed that the earliest Americans are ancestral to all living native groups. But ancient DNA work in the Cherokee homeland requires something no technology can provide on its own. It requires trust. Indigenous communities across North America have legitimate, deep, historical reasons to distrust genetic research. Decades of studies were conducted without informed consent. Samples were taken from ancestral remains without community permission. Results were published that contradicted cultural beliefs or were weaponized to challenge land claims. The Cherokee Nation, along with many tribal nations, now maintains strict protocols around genetic research involving their citizens and their ancestors. Any ancient DNA study in the Cherokee homeland must be conducted in genuine partnership with the tribal nation. Several such partnerships are reportedly in development. The technology to resolve whether the Cherokee arrived in Appalachia in a single large migration or through a gradual centuries-long process, and whether Mississippian cultural interactions reshaped Cherokee genetic diversity before European contact, exists right now. The sequencing platforms can extract usable DNA from remains that are thousands of years old. The statistical models can distinguish migration from admixture. The answer is within reach. And this is a side note, but it is worth mentioning because it connects to something larger. The explosion of commercial DNA testing over the past decade has flooded the public conversation with misunderstandings about Cherokee genetics. Millions of Americans have claimed Cherokee ancestry based on consumer test results that do not actually work that way. Those kits estimate regional ancestry percentages by comparing your DNA to reference panels, and the reference panels for Native American populations are notoriously thin. A consumer test cannot tell you if you are Cherokee. It can barely tell you if you are Native American. The real science, the work being done in partnership with tribal nations using controlled sampling and ancient DNA protocols, operates on an entirely different level of rigor. And it is that science, not the consumer kits, that is rewriting the Cherokee origin story. Right now, somewhere in the southern Appalachians, there are archaeological sites sitting quietly under layers of forest soil, sites that contain the physical evidence of one of North America's most significant prehistoric migrations. And somewhere in laboratories and tribal council offices, conversations are happening about whether and how to read that evidence. The Cherokee Nation today has over 450,000 citizens. It operates its own government, its own courts, its own language revitalization programs. And it is increasingly engaging with genetic science on its own terms, not as subjects of study, but as partners directing the research questions. The Cherokee have always known they came from the north. Their Kituwa stories said so. It took Western science 3,500 years of linguistic puzzles, 2 decades of mitochondrial mapping, and the invention of ancient DNA extraction to arrive at the same conclusion. The question now is not where they came from. It is what the journey looked like. And for the first time, the people whose ancestors made that journey are helping to lead the search.